Dopamine is synthesized from the amino acid tyrosine, which is obtained from food or supplements. Once tyrosine enters the bloodstream, it's transported into dopaminergic neurons by a carrier called L-type amino acid transporter 1. These neurons are found in several brain regions, most notably in the ventral tegmental area, substantia nigra, and the hypothalamus, just to name a few. The first step in dopamine production happens when tyrosine hydroxylase, with the help of its cofactors iron, oxygen, and tetrahydrobiopterin, converts tyrosine into L-dopa. Then, L-dopa is converted into dopamine by an enzyme called AADC, short for aromatic L-amino acid decarboxylase. This step removes a carboxyl group from the L-dopa molecule. For this to work, AADC needs a coenzyme called pyridoxal phosphate, the active form of vitamin B6. Finally, dopamine gets packaged into vesicles inside the neuron, ready to be released when needed. D1 receptors are one of the ways dopamine boosts brain activity. Think of them like the gas pedal in a car. When dopamine binds to D1 receptors, it tells the neuron, fire up. Here's how it works. When dopamine binds to a D1 receptor, it activates a G protein called GS, short for stimulatory G protein. GS proteins are like molecular switches sitting just beneath the surface of the cell membrane. They're made up of 3 subunits, alpha, beta, and gamma. In their inactive state, the alpha subunits are bound to GDP, guanosine diphosphate, a low-energy molecule. When the D1 receptor is activated, it triggers a shape change that causes the GS protein to release GDP and bind GTP, guanosine triphosphate, a high-energy molecule. This swap activates the alpha subunit, which breaks off from the beta and gamma subunits and moves along the inner membrane to stimulate adenylyl cyclase, an enzyme in the cell membrane. Adenylyl cyclase converts ATP, your cell's energy source, into cyclic adenosine monophosphate, or CAMP. CAMP acts as a secondary messenger, basically delivering dopamine's message into the cell. Once CAMP levels rise, it activates another key protein, protein kinase A. Protein kinase A is an enzyme that phosphorylates, adds phosphate groups to other proteins, including ion channels, cell transcription factors, and enzymes to change how the cell behaves. This increases the likelihood that the neuron will fire an electrical signal, reinforcing its activity and making it more responsive to future signals. Now here's where it gets especially important. D1 receptor signaling often works alongside glutamate, the brain's primary excitatory neurotransmitter. Glutamate binds to receptors like NMDA and AMPA on the same postsynaptic neurons. These receptors allow calcium and sodium ions to flow into the cell, initiating signaling cascades that contribute to synaptic plasticity, the brain's ability to form and strengthen neural connections. When dopamine via D1 receptors and glutamate via NMDA AMPA receptors are activated together, Especially in learning-rich environments, they potentiate each other's effects. Increased calcium ions from glutamate activity combined with protein kinase A activity from D1 signaling lead to increased transcription of genes related to memory formation, like C or EB, CAMP response element binding protein. CREB turns on genes that help neurons grow stronger, which is essential for long-term potentiation. This synergy, dopamine activating D1 receptors and CAMP protein kinase A pathways, and glutamate activating NMDA AMPA receptors, makes your brain actually learn and remember better. You don't just feel motivated. Your brain physically builds the memory faster and stronger. That's why when you're focused, hyped, or chasing a goal, your dopaminergic and glutaminergic systems are working hand in hand, sculpting your brain to be sharper, faster, and more driven. Now let's talk about D2 receptors. If D1 receptors are the gas pedal, D2 receptors are more like the brakes. Instead of exciting the neuron to fire more, D2 activation inhibits activity. But don't get it twisted, this doesn't make them any less important. In fact, D2 receptors are crucial for balance, precision, and emotional regulation. Here's how it works. D2 receptors are coupled to a different G protein called GI, which stands for inhibitory G protein. Just like GS proteins in D1 signaling, GI proteins sit beneath the cell membrane and are made of the same subunits. In the resting state, the alpha subunit is bound to GDP. When dopamine binds to a D2 receptor, the receptor activates GI by triggering the alpha subunit to swap GDP for GTP, just like with D1. But instead of activating adenylyl cyclase, GI inhibits it. That means less ATP is converted into CAMP and lower CAMP levels and protein kinase A activity drops. This causes downstream effects like less phosphorylation of ion channels, reduces the chance of neuron firing, less activation of gene transcription, and overall a common effect on neurons' activity. D2 receptors are also found presynaptically, meaning they sit on the neuron that's sending dopamine. These presynaptic D2 receptors act as autoinhibitory receptors. They detect how much dopamine is being released, and if it's too much, they signal the neuron to chill out and stop releasing more. It's like a built-in feedback system to prevent excitation and keep things balanced. D2 receptors play a big role in emotional regulation, impulse control, and motor skills. Dysfunction in D2 signaling is heavily implicated in disorders like schizophrenia, Parkinson's, and even addiction. While D1 and D2 receptors are the main stars of dopamine signaling, the D3, D4, and D5 receptors also play important roles, often more niche, but still crucial in fine-tuning brain activity. D3 receptors are part of the D2-like family, meaning they're inhibitory and also work through GI proteins, just like D2. So again, they inhibit adenylyl cyclase, reduce CAMP, and decrease neuron excitability. But unlike D2 receptors, D3 are found mostly in the limbic system. They're heavily involved in emotions, motivation, and addiction. Their role is more subtle. Think of them as a volume dial on dopamine's effects on mood and cravings. That's why some drugs that act on D3 are being researched for things like addiction, impulse control, and even schizophrenia. They influence how strongly we respond to dopamine-driven experiences. D4 receptors are also D2-like, so they share the GI pathway. But what makes D4 stand out is where it operates and what it connects with. D4 receptors are found in parts of the prefrontal cortex, amygdala, and hippocampus, areas tied to decision-making, attention, emotion, and memory. They're known to modulate both glutamate and GABA, helping balance excitation and inhibition. For example, in the prefrontal cortex, D4 activation can reduce glutamate release, helping keep neural activity under control. There's also strong evidence linking D4 receptors to ADHD. People with certain genetic variants of the D4 receptor may be more prone to attention-related issues. Interestingly, D4 receptors are also found outside the brain, included in the Leydig cells of the testes. These are the cells responsible for producing testosterone. Dopamine binding to D4 receptors on the Leydig cells can suppress testosterone production by inhibiting enzymes involved in steroid synthesis. It's a surprising crossover between brain and the reproductive system, showing just how far dopamine system really goes. Finally, the D5 receptor is like a cousin of the D1 receptor. It's excitatory, using the GS protein pathway just like D1, so it stimulates adenylyl cyclase, increases cAMP, and activates protein kinase A. The difference between the two: D5 receptors are less common and found in areas like the hippocampus and hypothalamus, where they likely help with learning, memory, and possibly hormonal regulation. Dopamine reuptake and breakdown. After dopamine is released into the synapse and does its job, it doesn't just hang around forever. It's quickly reabsorbed into the presynaptic neuron by a special protein called DAT, the dopamine transporter. Once it's back inside the neuron, enzymes like monoamine oxidase and COMT break it down. This stops the signal and keeps everything in a balance. So next time someone calls dopamine just a feel-good chemical, you'll know better. From how it's made to how it powers motivation, mood, and even memory, dopamine is way more than a buzzword. If you made it this far, your understanding of dopamine now goes deeper than most people will ever know. Believe in your ability to achieve great things, and thank you for watching.